Hi everyone, my name is Anya Lester, and I reviewed the article Under the Shadow: Old Biased Genes Are Subject to Weak Purifying Selection at Tissue and Cell Type Levels by Yildiz and a bunch of other researchers. And this was published around 2 months ago this year. For the background, I chose DNA because that pertains to their study. In the study, they reviewed the process of transcription and the RNA products that resulted in protein, and in turn, the protein that resulted in gene expression and how that correlated with age. Um, so they had a lot of hypotheses, one being that purifying selection in old biased genes would be weak, and another being that there would be a correlation in the conservation levels of the different types of tissue and different cell types during the aging process. And a third being um, something that relates to ADDICT patterns. And ADDICT stands for age-related decrease in transcriptome conservation, where a transcriptome is simply just the composite total sum of RNA in the genome of an individual in a species. The first hypothesis with ADDICT was that old biased genes can be subject to drift at the cellular level, which would decrease expression with age. And a second being weak conservation of transcriptomes are more visible in tissue at old age, resulting in increased expression with age. So within the study, they utilized the mutation accumulation hypothesis, which in class we discussed was the uh, purifying selection being weak in old age, resulting in deleterious mutations accumulating later in life, decreasing fitness and increasing drift. Um, Furthermore, they also wanted to see how the different tissue and cell types and conservation levels changed based on the species in the taxa in question that was observed. So they wanted to see if shifts in the conservation were due to change in tissue or cell, or if it was due to just change in organism, or if it had no effect. So they studied model organisms like the chicken, rat, mouse, killifish, and fruit fly, and they also used humans. To the right isn't something that was in the study, but it's just a phylogeny of the correlation of model organisms to humans to show why they're used. Within the experiment, they were very smart with how they collected their data. They utilized stuff that was already published, so they didn't have to do a lot more research than necessary, but they did their own calculations um, to create a composite transcriptome graph with respect to age. So they utilized the Spearman test, the Welch's t-test, and the non-synonymous synonymous mutation uh, correlation to create graphs. And then the results show that there were weaker conservation of old biased genes than young biased genes, and that depending on the cell type, there's also different levels of conservation. So immune cells were highly unconserved, while neural cells were very conserved. Immune cells were actually subject to inflammatory responses and apoptosis, which is programmed cell death, while neural cells had a process called proteotoxicity, which means like if they were to actually accumulate with mutations in the body, that would result in early death. And that's not something that would benefit the fitness of an organism, which is why neural cells are highly conserved. To the right is figure 3, and that just shows like the different tissue types and cell types and their conservation sequence determined by the Spearman test. And part A focuses on different cell types, while part B is specifically immune cells. So you can see immune cells are more unconserved than neural cells and um, muscle cells and stuff like that. So what was significant about this? Well, it proved the selection shadow phenomenon, which basically explained that harmful variants expressed at old age can't easily be removed due to the fact that um, after you're a certain age, you've already reproduced and passed on whatever's heritable to your offspring. So whatever shows up as heritable later in life is most likely already passed on. Um, that goes to trade-offs too, because when you increase fitness earlier in life and have high reproductive success earlier, obviously whatever's going to happen in old age is going to happen with the extrinsic factors and so on and so forth. So it also proved that transcriptomes are a very accurate and effective way of finding a correlation between species and between conservation statuses. Furthermore, this correlates to aging in metazoan species being pretty applicable and similar throughout the taxa. And metazoans are the vertebrates and invertebrates, the multicellular, multi-tissue plants and animals. Um, so because they had similar transcription or similar transcriptome levels and conservation statuses of each cell and cell type, um, It proved that it wasn't something unique to each species and kind of explain more about the process of aging. So it answers questions and raises questions at the same time, like what can be done about aging now? Yeah, thank you.